Look closely at these scales. Over the last few decades, selective breeding has turned this species into a highly organized system of human-directed visual design. The corn snake market is massive. Today, breeders have isolated over 40 distinct base mutations and documented more than 800 specific combinations. Premium prices are regularly paid for marketing names like palmetto or blood red, but these labels often overshadow the specific genetic code required to produce the animal's appearance. This creates a disconnect. Without understanding how these traits are inherited, specifically the mechanics of dominant versus recessive genes, a buyer is unable to distinguish between a standard visual trait and a high-value hidden carrier. Navigating this library of morphs requires a look at the inheritance types that dictate exactly what is being purchased. To understand the manipulation, we have to start with the biological baseline. The wild-type corn snake has a warm tan background with bold red-orange saddles bordered in black. This chart shows the three variables building that baseline: melanin for darks, erythrin for reds, and xanthan for yellows. Recessive mutations act as an off switch. Amelanism eliminates melanin entirely, leaving red and yellow on white. Turning off erythrin removes red, leaving cool dark grays. These single-gene morphs are a subtraction problem. Breeders are simply isolating the raw pigments that were always present by removing their competitors. The complexity shifts when breeders stack these recessive off-switches into a single animal. This logic gate shows the creation of a snow corn snake, combining amelanistic and aniristic mutations. By removing both black and red pigments simultaneously, the equation leaves the animal with virtually zero visible color, resulting in a ghostly white and pink appearance. Pattern-altering mutations add another layer to the math. These disrupt the spatial organization of pigment cells, turning standard saddles into stripes or random spots. Stacking 3, 4, or 5 of these color and pattern genes simultaneously allows for a massive range of distinct visual expressions. The diversity of the hobby comes from a predictable mathematical process of subtracting and rearranging genetic variables. While we can manipulate the color, there is a biological paradox here. Extreme modification of the exterior variables has zero impact on the internal physiology. Underneath the scales of a multi-gene mutant beats the exact same heart of a robust wild predator. Every corn snake requires 85 to 88 degrees of belly heat to function and digest food. Environmental requirements are equally rigid. The biological chassis requires a minimum 4 by 2 by 2 foot enclosure and humidity between 40 and 50%. We can play with the genetic math of the exterior, but the health of the animal depends on strictly maintaining the wild requirements at the center of the equation. To research specific varieties and their required care parameters, see the comprehensive corn snake morph directory at Krawlo.com.